Mirror neurons, the empathy cells that might not be introduction since the 1990s discovery of mirror neurons in macaque monkeys. Cells firing both when an animal performed an action and when it observed that same action. These neurons have been heralded as the biological substrate of empathy, imitation. Popular accounts trumpet mirror neurons as our brain's empathy cells. Do true mirror neurons even exist in human, might broader neural network? And why has the mirror neuron narrative captured the public imagination despite unresolved questions to move beyond the hype? We will explore 1. Controversies, 2. Alternative network explanations for imitation and empathy, and 3. The forces that turned mirror neurons into a pop culture neuroscience trope. 1. Do mirror neurons really exist in humans? Monkey origins versus human evidence in macaque single-cell recordings in area F5 and inferior parietal lobule IPL reliably identified mirror neurons. However, ethical and technical constraints prevent comparable single-cell studies in healthy humans. Instead, human mirror system research relies on indirect measures. fMRE and EEG findings. Functional MRI consistently shows that observing and performing actions activate overlapping regions. Similarly, mood rhythm suppression in EEG during action observation has been interpreted as mirror system engagement. Yet these population-level signals cannot resolve whether the same individual neurons or whether adjacent but distinct neuronal populations do direct recordings in patients. Rare opportunities arise when epilepsy patients undergo intracranial electrode implantation. Some studies report neurons in human promotor or parietal areas that fire for both action observation and execution, suggestive of mirror properties. However, sample sizes are small and response patterns vary. Many recorded neurons respond only during execution or only during observation, casting doubt on a robust human mirror cell population. Conceptual critiques. Critics argue that demonstrating single neurons with mirror properties in a few patients does not establish a dedicated mirror cell system. Instead, what appears as mirror activity might reflect task demands. While humans possess a mirror system of broadly overlapping activation, evidence for discrete mirror neurons analogous to those in monkeys. 2. Alternative network explanations for empathy and imitation. From single cells to distributed circuits, even if mirror neurons exist, they are unlikely to be the sole basis for complex social functions. Contemporary models emphasize network-level mechanisms, predictive coding framework. Under predictive coding, the brain constantly generates and updates models of action outcomes. This bidirectional exchange can account for action understanding and imitation without invoking special mirror cells. Any neurons, mentalizing versus mirroring empathy and theory of mind, engage a distinct mentalizing network. Medial prefrontal cortex, temporal parietal junction, and posterior superior temporal sulcus that infers others' beliefs and intention. Imitation tasks often recruit both mirroring and mentalizing systems, suggesting that understanding others' actions relies on integrating embodied simulation with higher-order inference rather than mirror cells alone. Hebbian learning and sensorimotor association. Repeated co-activation of sensory and motor representations during development could create sensorimotor link. Your own grasp circuits fire through learned associations, not because of a hardwired mirror cell blueprint, but via Hebbian strengthening of pathways connecting visual and motor neurons. These network perspectives paint social cognition as emerging from dynamic interactions across multiple brain systems. 3. The rise of a neuroscience pop culture trope. Why mirror neurons captured the public's imagination? Several factors propelled mirror neurons from niche discovery to neuroscience celebrity. Intuitive appeal. The idea that a single population of cells mirrors others' actions and feelings, simplifying complex brain functions into a memorable metaphor. Media amplification. Early high-profile publications and charismatic scientists framed mirror neurons and even autism therapies. Sensational headline. Educational and therapeutic promise mirror neurons. Inspired, enticing, but sometimes premature application. Mirror neuron-based interventions for autism, stroke rehabilitation through action observation, and social skills training predicated on enhancing mirror. Although some applications show potential, overselling mirror neurons has led to disillusionment when results prove variable metaphor over mechanism. Mirroring resonates as a metaphor for social connectedness, making it a favorite in self-help and management literature. Conclusion: mirror neurons remain a tantalizing but unresolved chapter in the neuroscience of social cognition. While humans exhibit mirroring at the level of brain region overlap and occasional single unit recall, the existence of dedicated empathy cells analogous to those in monkeys is far from settled. Broader network models, predictive coding, mentalizing circuits, and learned sensorimotor association offer richer, more flexible accounts of imitation and empathy, yet the mirror neuron tale endures in popular culture, thanks to its narrative power and intuitive clarity. Moving forward, scientists and communicators alike must balance engaging metaphors, ensuring that the next generation of social neuroscience stands on solid network-centered foundations rather than the fleeting allure of single-cell legends.